Hardworking enzyme keeps immune cells in line. LJI researchers foster new apparatus for understanding enzymatic action connected to blood tumors and coronary illness. LA Yola, CA researchers at La Jolla Institute for Immunology, LJI, have revealed insight into an interaction in resistant cells that may clarify why a few groups foster cardiovascular illnesses. Their exploration, distributed as of late in Genome Biology, shows the key job that TET catalysts play in keeping resistant cells on a soundtrack as they develop. The researchers tracked down the different chemicals to assume a part in this cycle, yet TET catalysts do the truly difficult work. On the off chance that we can sort out what's new with these chemicals, that could be significant for controlling cardiovascular infection, says Asushi Onodera, PhD, a postdoctoral analyst at LJI and first creator of the new Genome Biology study. LJI professor Anjana Rao, PhD, co-founded TET chemicals while working at Harvard University close by Mamta Tahiliani, PhD, and L. Arawind, PhD. Their work showed that this group of 3 compounds changes how our qualities are communicated. TET chemicals control quality articulation by setting off a cycle called demethylation, where an atom called a methyl bunch is taken out from where it sits in the hereditary code. Demethylation is significant on the grounds that it's anything but a cell, peruses DNA. Throughout the last decade, Rao has shown the significance of TET action in malignancy improvement. Her work has uncovered that TET compounds are vital to appropriate quality articulation in resistant cells, and they can really ensure against harmful transformations. For the new examination, Rao and Onodera researched how invulnerable cell DNA can be adjusted by either TET chemicals, a cycle called aloof demethylation, or by a DNA fix catalyst called TDG, dynamic demethylation. The scientists meant to uncover which demethylation pathway has a greater job in deciding the quality articulation, the very destiny, of insusceptible cells. The specialists began with 2 resistant cell models, CD4, AID, T cells and monocytes. Both cell types should multiply and develop into more explicit cell types to assist battle with offing microorganisms. Notwithstanding, whenever monocytes are separated into macrophages and animated with a particle called LPS, they quit multiplying. By investigating these CD4 assistant T cells and macrophages, the scientists could more readily comprehend multiplying and non-multiplying models. The multiplication interaction is extremely speedy, making it an ideal opportunity to observe what demethylation happens and how it means for quality articulation. Onodera utilized CD4 assistant T cells to investigate the demethylation cycle utilizing a state-of-the-art computational examination program created for this examination. This device gives researchers a remarkable glance at what areas of DNA inside a cell are methylated. We tracked down that in insusceptible cells, most demethylation occurs through the uninvolved pathway, clarifies Onodera. Utilizing another method called pyridine-borane sequencing, the analyst showed that dynamic demethylation through TDG is working in invulnerable cells. Onodera says TDG's job is minor. It does the work of eliminating 2 atoms created by TET chemical action. This revelation is significant in light of the fact that TET changes can be dangerous. Past examinations have shown that transformations in the TET2 protein handicap, or meddle with, a portion of the ordinary limitations on solid monocytes. The monocytes denounce any and all authority and develop into provocative macrophages. Individuals bearing TET2 transformations in macrophages have a 40% increment in their danger of creating cardiovascular infection. A more profound comprehension of how TET chemicals work could help analysts realize where to intercede and assist patients with this sort of transformation. Onodera's new investigation program for recognizing changes in DNA adjustment may likewise demonstrate helpful for seeing how TET transformations in resistant cells can prompt blood malignant growths. He clarifies that scientists might actually utilize the program to distinguish patients who may have distinctive treatment results dependent on where their DNA has been demethylated. Ideally, we can apply this method to illness analyze, says Onodera. Truth be told, Onodera says the new sequencing and examination program from this investigation can be applied to numerous invulnerable cell types and numerous sickness models. Now, we can gain critical headway in understanding the subatomic instruments behind DNA demethylation, Onodera says.